Hello, my name is Geoff MacKay, President and CEO of Avrobio, and I'm very pleased to update you on our very solid progress that we've made over the last few months. Before we begin, the presentation will contain forward-looking statements based on our current beliefs and expectations, and I refer you to our SEC filings for further information. We recently celebrated our 5th birthday, and as a company, our founding purpose hasn't changed, which is to deliver freedom from a lifetime of genetic disease. The data that I'll show you highlights that we're on track to do just that. Our goal is targeting lysosomal disorders to not only untether patients from biweekly infusions, but ideally to halt disease progression. Now, there's not enough time to cover our complete data set, but I'll lay out firstly our approach, followed by some of the more meaningful data that we've recently shared. First of all, our pipeline. And what you see is 6 lysosomal storage disorders, so essentially sister indications with essentially common underlying biology. And what's important about that is having already demonstrated proof of concept in Fabry disease, we believe that it, at least to some extent, de-risks or changes the probability of success of the sister indications. Very congruent set of indications, and also indications that cost the healthcare system a fortune. Fabry, for example, has a lifetime cost of direct expenses, meaning the medicine itself, of anywhere from $12 to $14 million. The original pioneers in this field, Shire and Genzyme, even today, now Sanofi and Takeda, sell almost $4 or $5 billion of standard of care. However, there is a significant unmet need that remains. When you think of lentiviral gene therapy, the one word that comes to mind is durability, and that's because of the integrating nature of the vector. That's been demonstrated in hundreds of patients across a dozen different indications, and now it's also demonstrated by us. What you see here are all of our patients with the duration of how far we followed them post single infusion of gene therapy. And what you see now is it's starting to become meaningful with several patients out a year or 2, and the furthest patient out 3.5 years without waning of effect. Now, we at AVRO have had a total commitment to CMC manufacturing. And of course, there have been a number of pretty material delays in our field in the last year or so. So I really wanted to highlight some of the things that we're really the most proud about. And these are some of the brag points, is that what we're trying to do is to take a very manual Petri dish type science and turn it into a scalable pharmaceutical grade medicine. For example, to our knowledge, we're the only company that is operating in an automated closed system on 3 continents. We are one of only a few groups that have scaled up our vector such that we manufacture our lentiviral vector in a 200-liter serum-free suspension bioreactor. And with a 10 to the 9 titer, that means every single run is enough to treat 40 or 50 patients. And in addition, we've optimized each component. For example, the vector, pivoting from what we now call our academic 3-plasmid lentiviral vector system to a much more state-of-the-art tuned 4-plasmid lentiviral system that's really been designed for potency, safety, and manufacturability. And then a final key component of this set of steps is the final step prior to infusion, which is conditioning. And AVRO has also invested in pivoting from a low-dose malfilin approach to what we call busulfan or Bu90 target concentration intervention, designed to, first of all, optimize the distribution of the gene therapy head to toe, but also significantly improve various safety parameters. So to jump into the data, what I'll start with are our first wave programs where we actually have human clinical data to share: Fabry disease, Gaucher disease type 1, and cystinosis. Starting with Fabry disease, there are about 10,000 patients. The world tends to know of these diseases because of the pioneering work of companies like Shire and Genzyme. And they used to be thought of as kidney and cardiac diseases, but really they impact the patient head to toe every day of their life. ranging from, of course, kidney and cardiac, but their fingers feel like they're on fire, tremendous GI issues. And about 50% of these patients have tremendous white matter lesions, which translate into executive function deficit, what the Fabry community calls the Fabry fog. So a very serious disease, and our target product profile is to deliver enzyme head to toe and therefore address the symptoms that manifest head to toe. Probably the most exciting data that I have to share today is the primary endpoint of our global phase 2 trial, looking at classic male Fabry patients that have never received enzyme replacement therapy, so ERT naive. And what you see in the first 2 evaluable patients is essentially a complete eradication of the substrate GB3, meaning the average number of GB3 inclusions per peritubular capillary. So this kidney function is really remarkable and compares very favorably to many modalities that have studied this very same endpoint. It's also of particular importance because this endpoint is referenced in the FDA Fabry guidance documents as acceptable for accelerated approval. So we couldn't be more thrilled to have a net 93% reduction of substrate in the kidney in the first 2 patients measured. Now, behind that striking kidney data is a lot of biomarker data. And of course, there's been 40 years of science and 20 years of clinical experience in this indication. So a lot is known. And of course, the biology isn't that complicated. Enzyme up, substrate down. So the first thing we want to show is relevant sustained expression of enzyme alpha-galactosidase A in both the plasma and the leukocytes. And that's exactly what we see across all phase 2 patients. The patient that is really operating in a different postal code in black is our first Plato patient. So the first patient that is leveraging now our new commercial stage optimized platform. On the bottom, you see vector copy number trending exactly as we would hope, reaching a plateau and staying steady on that plateau across all patients. And as a result of enzyme up, what we see in these classic male Fabry patients is substrate down. Here, we're looking at plasma lyso-GB3 reduction, which is the toxic metabolite. I would note in blue that that patient is actually a cardiac variant, so a different form of Fabry, and those patients don't manifest with lyso-GB3 in the plasma. So we just include for transparency. But in the evaluable patients, an average of 70% reduction, very notable. Now, if we jump to our phase 1 program, which is complete, fully recruited, with the furthest patient out 3.5 years, this is a different group where we can get different insights. These patients have had biweekly infusions of enzyme ERT for years and years leading into the study. They were deemed ERT stable. And so now we can ask the important question in a sort of within patient control methodology, how does gene therapy perform relative to ERT baseline? And the few quick statements that we can make is that not only is gene therapy controlling these patients, but there's a further reduction, a 25% further reduction of the toxic metabolite, plasma lyso-GB3, when gene therapy is added. And importantly, all patients who discontinued ERT remain off ERT. And then the final point, going out as far as 3.5 years, there is no waning of effect over time across patients. So we're very pleased with the phase 1 efficacy data. Now, as we aggregate more patients with 9 total across both studies and more time, we can start to look at important functional measures. And one of the key ones in this disease is how is the kidney doing? And so we look at eGFR. And again, there's a richness in natural history in Fabry disease. So we understand the steady annual decline of eGFR in published natural history and in published ERT studies. And what we see is a different slope, meaning 8 of the 9 patients are not declining anywhere near the slope as well documented in natural history. And even the 9th patient, the one on the bottom who starts with significant kidney disease in eGFR of 48, does decline over a few years, but at a slope that is different, better even than natural history. for a patient that's entered the study with such significant pre-existing disease. So more time will derive more value from looking at these longer term functional measures. But what we can say already is the slope compares very favorably to natural history and ERT studies. And similarly, we look at cardiac function. And what we see is the patients in the phase 2 entered the trial within the normal range and stayed within the normal range. So that's about as good as we could hope, but the value again will come in longer term follow-up. So far so good on all cardiac measures. Now, we're really excited about the efficacy, the durability, but everything in life is risk benefit. So the important question is, what about safety? What about patient tolerability? And we are not seeing AEs or SAEs linked to the gene therapy. What we do see are predictable adverse events associated with a concomitant conditioning regimen. And I mentioned that we, as part of Plato, pivoted to what we think is a better and optimized conditioning regimen. What we see is this Bu90 TCI approach in magenta with our original low-dose malfolin approach in the sort of gray-blue. So importantly, the adverse events that we're seeing with our optimized conditioning are more finite in nature, transient. They tend to arrive and depart in week 2 following conditioning and are quite predictable. So we are very satisfied with the profile that we're seeing today. Now, if I jump to our second indication, cystinosis. Cystinosis is a little bit less well known. There's about 2,000 patients in the world, very serious. Cysteamine is the standard of care, but even on cysteamine, 90% of these kids will get a kidney transplant, severe visual impairment, failure to thrive, CNS impact, a tough disease. So we're really excited to see if gene therapy can play a material role in improving their care. We've dosed 3 patients to date, and the first thing we can say is all 3 patients immediately stopped their 30 pills of cysteamine a day, which in itself has a tremendous benefit to the patients because the standard of care is so poorly tolerated. Patient number 1, for example, went from vomiting 13 times a day to vomiting 0 times a day in the first 3 months of gene therapy, attributable to getting off cysteamine. And I mentioned 90% of these kids have kidney transplant. Patient 1 made it to the age of 20 without a kidney transplant, but what you can see is where he's headed. You can see the sharp, steady decline of eGFR, and it's too early to make claims, but we're certainly satisfied and in fact excited at the change of slope of eGFR following the discontinuation of cysteamine and the uptake of gene therapy, the single infusion of gene therapy. Now, we're looking at many, many measures across the patient, taking skin biopsies, rectal biopsies, and across both measures, we're not only seeing a very significant reduction of crystals that form in the cells, but also the volume of those crystals. So really nice data in patient number 1, looking at baseline versus 12 months following that single infusion. But even beyond skin and rectal, something that is very important to these patients is their vision. And as I mentioned, vision is one of the things that they really struggle with, severe photophobia. And this slide shows why. In blue is the baseline view of the patient's cornea. And you can actually see this real time without a microscope. Crystals accumulate in the cornea, leading to that severe photophobia. And as a result, the patients are expected to take 8 hourly eye drops of cysteamine a day. And either that is incredibly onerous or they just don't for compliance reasons. But here you see 12 months after gene therapy, not a single eye drop during that time period, and not a complete resolution, but darn close. What this has translated to the patient is now that patient, rather than having to wear sunglasses, dark glasses, leaving the house, can leave the house without sunglasses at all. And prior to being unable to even check his phone at night because of the pain from the light, like the rest of us now, he can check his iPhone at 2 in the morning. So a really dramatic change in the quality of life on just this one functional measure. And I'll conclude cystinosis with a really, I'd say, wow factor that hopefully will turn on a light bulb about the power of gene therapy that can be extrapolated beyond cystinosis, is we understand that proteins are multifunctional. That's how evolution made us. And it just so happens that the cystinosin protein regulates melanin synthesis as a secondary or tertiary measure. It's called moonlighting proteins. And typically proteins do have functions that many of which we don't even understand. But here, these cystinosis patients are very typically pale, lacking pigment, much like patient number 1 on the left. And after a single infusion, in a matter of months, his physical features began to transform. And now, to quote him, he looks for the first time in his life like his brother. His family has darker pigment. So this really, I would invite you to think about on other gene therapy applications. Fabry, for example, we're very narrowly looking at enzyme up, substrate down. In all likelihood, this gene augmentation approach is having benefits that we have to challenge ourselves or invite ourselves to begin to measure. And of course, the safety profile in line with expectations, no adverse events related to the gene therapy, standard predictable adverse events related to the concomitant conditioning regimen. Final indication is Gaucher type 1. Rather than kidney or cardiac, this tends to be liver, spleen, bone, but like Fabry, a head-to-toe disease. There's about 15,000 Gaucher type 1 patients in the world. So we'll look at the first patient to receive a gene therapy for Gaucher type 1. This was a 32-year-old female patient who started enzyme replacement therapy at the age of 3. And this is just 10 years ago when, as is absolutely par for the course, she went through a period of non-compliance. And you saw one of the key biomarkers, chitotriosidase, go from 150 to spike up into the thousands. So now, if you advance the clock several years, let's see that same patient also stopping ERT, but this time being put on gene therapy. And on the right, again, chitotriosidase, the same biomarker. And what you see, a starting point of 151, 7 months after stopping ERT, not only is it controlled, but it's controlled 49% better than baseline VPRIV, the Shire Takeda product, the enzyme replacement therapy. And another absolutely valid biomarker is the toxic metabolite itself, lyso-Gb1, also showing improvement on gene therapy alone versus baseline VPRIV. So we're pretty excited about that. Other relevant measures in Gaucher type 1 is, can you keep platelet counts and hemoglobins within the normal range? And the quick answer is, in patient number 1 out 6 months, yes, all measures remain within the normal range. And then in terms of safety, very similar to Fabry, very similar to cystinosis, is that applying our Bu90 TCI, a single agent, single dose, mid-range level with tight area under the curve monitoring, has been able to take the traditionally toxic bone marrow transplant application and ideally harness it such that the adverse events we're seeing are largely contained to the second week following infusion of gene therapy, where they come as platelets and neutrophils drop to a low nadir, the neutrophils and platelets bounce up, and the adverse events tend to be resolved. So we're, again, pleased with what we're seeing, and we hope we can continue to replicate this value proposition with this type of adverse event profile. So with that, I'll thank you for your attention and really look forward to continuing to progress our clinical stage gene therapies and updating you at the next possible opportunity. Thanks again.